Now that you've had a chance to look at one trait in a variety of different contexts, we are going to be looking at two traits at the same time and trying to predict the genotypic and phenotypic ratio of offspring based on those dihybrid traits. So here is an example. We have two purebred or true-bred cats at the top here. These will be the two parents. Notice, because they are homozygous in two traits, there's only one possibility as far as what sort of gamete they will have. And so we have our female cat on the left and our tomcat here on the right. And when they crossbreed, all of the first generation here are now heterozygous for both traits that we have here. Note the traits in question are symbolized with SS, the big S being a short tail, the little s being a long tail. And then we also have BB, with the big B being a brown coat and the little b being a white coat. So what's going to happen when these parents have their offspring? Well, each of heterozygous parents have four possible combinations of alleles that they can make. And this goes back to using kind of our FOIL method. You can have big S, big B. You can have a big S, little b, a little s, big B, and a little s, little b. And so there are four different possible combinations that we have here from each of the parents. And notice we have the female on this side and the male there on the top. And so with those four different gametes and four different gametes for the male and four different gametes for the female, that leads to 16 different possible combinations that can happen. So just to look at the first one here, for instance, we've got an S and a B from the father, and there's the big S and the big B there, and an S and a B from the mother, and there's the big S and big B there. And so now they are homozygous dominant for both traits. We can skip down the line here, and for instance, if you look at this, and we've got a little s and a big B, and we go back to our traits from the mother here, big S, big B. So here's the little s from the father, and here's a big B from the father, and there's a big S from the mother, and a big B from the mother. So the first trick is to really do these successfully, is you have to look at what possible gametes you can do there. You can do your foiling, your first, well, in this case they did outer, inner, and last, to get the possible gametes that can occur from the parents. Once you have the gametes from each of the parents, the possible gametes, then it's just a matter of filling in each of these boxes by dropping them down just like we've done in Punnett squares before. Now, one of the main things that we start to notice here is that there tends to be a typical sort of ratio that appears, phenotypic ratio at least, and that would be 9 to 3 to 3 to 1, with 9 being dominant for both traits, 1 being recessive for both traits, and then you've got your 3 here that are dominant with the short tail and recessive with the coat, so dominant for one trait and recessive for the other. And then you have your 3 here that have the long tail, which is recessive, but the dominant brown coat and recessive, and then dominant for the two traits. And so as we look at phenotypes, that's what we tend to get there. So there is a quick summary of how to do a dihybrid cross. One thing to keep in mind is this is the most complicated dihybrid cross you can have when both parents are heterozygous. We've got that phenotypic ratio of 9 to 3 to 3 to 1, but if we happen to have a parent that is not heterozygous for both traits, it makes things a little bit easier, like we had talked about before. So for instance, this parent here on the left is going to have to give a big S gene, because that's all they have. So we can simplify it. Instead of having all 16 boxes, we only draw 8, and it makes it much simpler. And so in this case, you can see our phenotypic ratio. There are no long-tail cats because there's no way for any of the offspring to get 2 little s's. So our phenotypic ratio is 3:1, which should look kind of familiar because that's the phenotypic ratio you'd normally get for normal dominant recessive trait when you just had a monohybrid cross. So in summary, the first thing is to figure out what possible zygotes can you get from the parents. And so in the case of this one, they have to get a big S, and then they can either have a big B or a little b. In the case of this one, they can have a big S or a little s. They can have a big B or a little b. And so there's 4 possible gametes from that parent, but only 2 from the first one. And so once you have those, you make your Punnett square, and then the rest is the same as we've done before.